Simulation of audition begins when a compression of air between 15,000 and 20,000 hertz of air is collected by the outer ear, or the pinna. The air travels down the ear canal until it hits the tympanic membrane, turning the air into vibrations. The vibrations travel through the malleus, the incus, and the stapes until these vibrations hit the oval window of the cochlea. The transduction of audition begins when the vibrations of the oval window reach the fluid within the cochlea. The movement of fluid causes the hair cells, which are between the basilar membrane and the tectorial membrane, to move. These hair cells are also called auditory receptors. Transduction continues as the hair movement excites the auditory nerve. Once the auditory nerve has been excited, signals are sent ipsilaterally to the superior olivary nuclei. Fibers from the superior olivary nuclei ascend to the inferior colliculi. The axons of the inferior colliculi ascend to the medial geniculate nuclei. Axons projected, which are also called auditory radiations, reach the primary auditory cortex. The stimulation of gustation begins when chemicals are perceived as sucrose, hydrochloride, sodium chloride, and umami. These chemicals first interact with the papillae. On the papillae are taste buds. The taste buds contain taste receptors. The excitable membranes of the taste receptors help to release the neurotransmitter to excite the neighboring cells. This begins the transduction. Axons of the taste receptors collectively compose the facial nerves. The facial nerve enters the NTS, ascending towards the brain. The NTS branches out to the VPN of the thalamus, as well as other areas of the brain like the medulla and the amygdala, and areas of the cerebral cortex. Of course, the main cortex associated with the facial nerves in relation to taste is the primary taste cortex. Stimulation of olfaction begins when odorants enter the nasal cavity, the nose and the sinus cavity. These odorants are received as chemicals on the cilia of the olfactory receptor cells. Information from the chemicals is passed through the olfactory nerve axons, which synapse at the mitral cell, which are located in the olfactory bulb. the axons of the mitral cells in the olfactory bulb become the facial nerve that will ascend to the brain. The transmission pathway for olfaction is the direct pathway of the mitral cell axons to the olfactory cortex. The stimulus for the somatosensory touch begins with movement of hairs on the skin surface, sudden displacement of skin, light touch, stretching of the skin, and stretch or injury to joints and muscles. All of these stimulate receptors associated with these movements. These touch receptors are free nerve endings, hair follicle receptors, messenger corpuscles, Pacinian corpuscles, Merkel's discs, Ruffini endings, and cross end bulbs. All of these touch receptors become stimulated, and when they become stimulated, sodium channels open and an action potential begins. Following the transduction of touch, transmission begins. Transmission starts when the information from receptors below the head enter the spinal cord via dermatomes. Dermatomes are areas of skin associated with a spinal nerve. The information enters the dorsal root from the spinal cord. Then ascending ipsilaterally to the dorsal column nuclei. Input from the leg synapses at the nucleus gracile, and input from the arm synapses at the nucleus cunatus. Decussitation of the transmission pathway occurs via the axons of the dorsal column nuclei. The axons continue to ascend by way of the dorsal column medial lemniscus until synapsing at the ventral posterior nucleus, the VPN, which is in the thalamus. Axons from the VPN project to the primary sensory cortex. The somatosensory cortex has a map of the body on it, which is also called the homunculus. Stimulation of somatosensory pain begins when primary sensory nerves register pain, have pain receptors, and which begin to synapse in the dorsal root of the spinal cord after following an action potential. Decussitation of the transmission pathway takes place as the secondary neurons ascend via the lateral spinal thalamic tract. The secondary neurons then synapse in the VPN of the thalamus. Tertiary neurons ascend to the primary sensory cortex. In addition to ascending to the primary sensory cortex, these neurons also branch off to the limbic system and even the amygdala. In these areas, pain isn't perceived, but emotional associations are sensed instead. Lastly, certain kinds of painful stimuli send emotional pain information from the sensory cortex to the periaqueductal gray. This information is then transmitted through endorphins. The axons from the PAG reach to the back part of the medulla, thus exciting cells which release more endorphin neurotransmitters to the spinal cord. Although pain is still being received via the dorsal roots of the spinal cord, the endorphin has inhibited the primary sensory nerve, which is releasing substance P, therefore creating a centrifugal pathway, which stops areas of the spinal cord from receiving pain messages.